Biochemistry, molecular genetics. Molecular genetics is a branch of genetics that studies the structure, function, and regulation of genes at the molecular level. Specifically, molecular genetics examines how genetic information is stored, maintained, and inherited in the form of DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, and how this information is used for the synthesis of proteins that underlie biological functions in cells. The following is a detailed explanation of the main concepts and processes in molecular genetics, DNA structure and function. DNA is the main genetic material that stores information for the formation and regulation of all biological processes in organisms. DNA consists of two polynucleotide chains that form a double helix structure. DNA structure components, namely, nucleotides, namely, the basic unit of DNA consisting of three main parts, namely, 1. Deoxyribose sugar, pentose or 5-carbon sugar. 2. Phosphate group. 3. Nitrogen base. Where there are 4 types of nitrogen bases in DNA, namely, 1. Adenine, A2. Thymine, T3. Cytosine, C4. Guanine, G, base bonds, namely, these bases pair with specific rules, namely, adenine pairs with thymine, and cytosine pairs with guanine, through hydrogen bonds. Double helix, namely, 2 DNA polynucleotide chains twist together to form a very stable double helix structure and allow for tight storage of genetic information. DNA functions, namely, DNA is responsible for storing genetic information used in the formation of RNA and proteins needed for cell structure and function. 2. DNA replication. DNA replication is the process by which DNA molecules are duplicated to produce 2 identical copies of genetic information that will be inherited to daughter cells during cell division. DNA replication process, divided into 1. Helix opening, namely, the helicase enzyme opens the double helix by breaking the hydrogen bonds between base pairs. 2. New chain synthesis, namely, the DNA polymerase enzyme adds nucleotides to the new chain based on the principle of base matching, A with T, C with G, on each template chain. 3. Formation of 2 DNA molecules. Namely, each strand of DNA is used as a template to form a new strand, producing 2 identical DNA molecules. DNA replication is very important to ensure that the genetic information contained in DNA is passed on accurately during cell division. 3. Transcription, namely, formation of RNA from DNA. Transcription is the process by which the information contained in genes in DNA is copied into RNA molecules, specifically messenger RNA, mRNA. which carry genetic information for protein synthesis. Transcription process, namely 1. RNA polymerase binding, namely RNA polymerase enzyme binds to the promoter region at the beginning of the gene, starting transcription. 2. RNA synthesis, namely RNA polymerase moves along the DNA chain and synthesizes mRNA by combining RNA nucleotides A, U, C, G, based on base matching rules similar to DNA, but T is replaced with U, uracil. 3. End of transcription, namely when RNA polymerase reaches the terminator signal, the transcription process stops and the newly synthesized mRNA is released. The mRNA produced will carry genetic information from the cell nucleus to the ribosomes in the cytoplasm for the next process, namely translation. IV. Translation process, namely protein synthesis. Translation is the process in which mRNA that has been copied from DNA is translated into a sequence of amino acids to form proteins. Translation process, divided into 1. Initiation, namely the process begins when mRNA binds to ribosomes in the cytoplasm. Ribosomes read the genetic code in mRNA in the form of base triplets called codons, 3 bases representing 1 amino acid. 2. Amino acid selection, namely tRNA, transfer RNA, carries amino acids that match the mRNA codon. Each tRNA has an anticodon that pairs with the codon on the mRNA. 3. Elongation, namely, ribosomes move along the mRNA, reading the codons one by one, and tRNA carries the appropriate amino acids to form a polypeptide chain. 4. Termination, namely, the translation process ends when the ribosome reaches a stop codon, e.g. UAA, UAG, or UGA, which signals the end of protein synthesis. The newly synthesized protein is released from the ribosome and can be folded into its functional form. V. Genetic mutations. Mutations are changes in the DNA base sequence that can occur naturally or be caused by external factors such as radiation, chemicals, or viral infections. Mutations can affect protein synthesis and can have a major impact on cell function. Types of mutations, including 1. Point mutations, namely, changes in one DNA base pair, e.g., base substitution, where one base is replaced by another base. 2. Insertion or deletion mutations, namely, addition or removal of one or more nucleotides in the DNA sequence, which can change the codon reading frame and produce defective proteins. 3. Silent mutations, namely, mutations that do not change the encoded amino acids, due to redundancy in the genetic code. 4. Stop mutations, namely, mutations that produce premature stop codons, which can affect the length and functionality of the protein. Mutations can be beneficial, neutral, or detrimental. Detrimental mutations can cause genetic diseases or cancer. V. Regulation of gene expression. Gene expression is the process of controlling the level at which certain genes are expressed to produce RNA and proteins. It involves a variety of mechanisms that allow cells to regulate when, where, and how much protein is made, including 1. Regulation of gene expression in prokaryotes. In bacteria, regulation of gene expression is often accomplished using operons, such as the lac operon in Escherichia coli, which controls the expression of genes involved in lactose metabolism. This regulation usually involves binding to control elements, such as inhibitors and activators, that control gene transcription. 2. Regulation of gene expression in eukaryotes. In eukaryotic cells, regulation of gene expression is more complex and involves more control elements, including enhancers, silencers, and transcription factors that bind to promoters or other regions in the DNA to activate or deactivate transcription. DNA methylation and histone modifications also play important roles in regulating gene expression. V. Genetic recombination. Genetic recombination is the process of exchanging genetic information between DNA molecules, which can result in genetic variation. In the process of meiosis, genetic recombination allows the exchange of chromosome segments between homologous chromosomes, which increases genetic diversity in offspring. 8. Techniques in molecular genetics. Molecular genetics uses a variety of laboratory techniques to manipulate and analyze DNA and RNA, including 1. PCR, polymerase chain reaction, namely, a technique for amplifying or multiplying small segments of DNA. 2. Gel electrophoresis, namely, used to separate DNA molecules based on their size and electrical charge. 3. DNA sequencing, namely, a technique for determining the sequence of DNA bases accurately. 4. DNA recombination, namely, combining pieces of DNA from different sources to create recombinants, for example, in the production of recombinant insulin. Conclusion: molecular genetics is the study of how genetic information is stored, expressed, and inherited in the form of molecules such as DNA and RNA. Understanding processes such as replication, transcription, translation, and regulation of gene expression allows us to understand how the body functions and how genetic variation can affect health and disease. This knowledge also paves the way for the development of genetic therapy, biotechnology, and more personalized medicine.